Hello everyone, my name is Zaidatul Shakila binti Muhammad Ashari, and today I will continue NC3, C4 and CAM plants. Let's get started. In this video, I'm going to show you the alternative mechanism on carbon fixation in C4 and CAM plants, and then explain what are the steps involved in C4 and CAM pathway, and then differentiate between C3, C4 and CAM plants. Recall that photorespiration occurs due to the accumulation of oxygen molecules and decreased carbon dioxide molecules that results as stomata closed. Plants that live in a hot climate, they are at risk to photorespiration, and they have evolved method to avoid photorespiration by maintaining high concentration of carbon dioxide. In order to minimize photorespiration, plants need to increase the concentration of carbon dioxide so that Rubisco enzyme is less likely to produce glycolate through reaction or combination with the oxygen. There are two groups of plants that have evolved to minimize photorespiration. First group that achieve this by maintaining high carbon dioxide level as a result of structural rearrangement in the leaf. Plants with this structural rearrangement or hatch and slack pathway in leaf known as C4 plants. This plant fixes carbon dioxide at two different cells. One is mesophyll cell and another one is bundle sheath cells. It captures carbon dioxide in mesophyll cell first and then release carbon dioxide into the bundle sheath cell. And then second group of plant that evolved to minimize photorespiration by maintaining high carbon dioxide level use stored carbon dioxide in the form of organic acids. This plant known as CAM plant. This plant fixes carbon dioxide at different time during night and during day. During night, it allows absorption of carbon dioxide to take place so that the stomata can be closed during the day to minimize water loss. We will examine what happened in C4 plant first. Example of C4 plants are corn, sugarcane, sorghum or crabgrasses. C4 plant conduct C4 photosynthesis, and why it is called C4? Just a reminder, in classic Calvin cycle, the first byproduct is the 3-phosphoglycerate. And 3-phosphoglycerate contains 3 carbon atoms. So we call Calvin cycle as C3 photosynthesis. And the plant that conducts C3 photosynthesis is C3 plant. Okay, it is named after the very first product produced when there is a very first time carbon dioxide fixation process occur. So in C4 plant, the very first time carbon dioxide fixation will produce a 4 carbon atoms molecule. Thus, this C4 plant undergoes C4 photosynthesis. So C4 plants conduct C4 photosynthesis in mesophyll cell, and then Calvin cycle or C3 photosynthesis occur in the bundle sheath cell. Remember that I told you earlier that this kind of plant, C4 plant, has a structural rearrangement. Two cells involved in order to increase photosynthesis rate and minimize the cost of photorespiration. So C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into 3 carbon compounds, which is named as phosphoenol pyruvate, PEP. Okay, you have heard PEP or phosphoenol pyruvate in glycolysis chapter 2. Alright, in mesophyll cells, and produce a 4 carbon compound named oxaloacetate. This step requires the enzyme named PEP carboxylase, phosphoenol pyruvate carboxylase. PEP carboxylase has a higher affinity for carbon dioxide than Rubisco does. It can fix carbon dioxide when carbon dioxide concentration is low. These oxaloacetates are forming the malate, and then malate exported to bundle sheath cell where they release carbon dioxide that is then used in the Calvin cycle. Okay, what about the chloroplast distribution in C3 and C4 plants? Is the arrangement of the leaf structure same or different? Let's recall the cross section of C3 plant leaf. The mesophyll cells of C3 plant is between the upper and lower layer of epidermis cells. The layer of mesophyll cells consists of tightly packed cell and loosely packed cells. The bundle sheath cell has a direct access to the air due to the presence of space here within the loosely packed cells. Now let's take a look on cross section of C4 plant leaf. Remember what I have said earlier, C4 plant maintaining high concentration carbon dioxide as a result of structural rearrangement in the leaf. Okay, like the leaf of C3 plants, C4 leaf has also the upper and lower layer of epidermis cells and the stomata at the lower layer of epidermis cells. The different structural arises in the mesophyll cells layer, in which the vein, aka the vascular tissue in the center, is wrapped by the bundle sheath cell, and then the bundle sheath cells wrapped by the mesophyll cells. See here, the bundle sheath cells have no direct access to the air, and then this structure of leaf referred to Kranz anatomy, whereby the mesophyll cells is arranged in a circle around bundle sheath cell. This structure is to ensure that the organic acid can easily pass to bundle sheath cell from mesophyll cell. How the structural rearrangement of C4 plant leaf can reduce the photorespiration? Use zoom in picture from vein, the vascular tissue, bundle sheath cell and mesophyll cell of C4 leaf anatomy to explain the process. Recall from all photorespiration discussion, plant in hot and dry environment has high concentration of oxygen and low concentration of carbon dioxide in leaf due to the closure of stomata. So within the mesophyll cell, an enzyme called PEP carboxylase has a higher affinity for low concentration of carbon dioxide rather than oxygen. So even in the condition whereby the ratio of oxygen is higher than carbon dioxide, the PEP carboxylase will pick off carbon dioxide and attach it to the PEP molecule. So this results form a molecule named oxaloacetate. Oxaloacetate then converted to malate. Malate enter bundle sheath cell and release carbon dioxide and then become pyruvate. The pyruvate re-enter the mesophyll cell and reconverted back to PEP with energy input of ATP. So the purpose of this pathway is to concentrate the carbon dioxide within the bundle sheath cell, to increase the concentration of carbon dioxide in bundle sheath cell. So this pathway is called C4 pathway or C4 photosynthesis. Next, the Calvin cycle occurs in the bundle sheath cell of C4 plant because carbon dioxide is highly concentrated in this cell. The carbon dioxide in the bundle sheath cell attached to the RUBP by Rubisco enzyme. Okay, this is the classic Calvin cycle as usual. And then the sugar, which is the G3P produced from the Calvin cycle, is transported into the vascular tissue. Okay, let's recall the C4 pathway in C4 plant until it produces the sugar. See the numbered steps in the figure. So number 1, phosphoenol pyruvate aka PEP that contain 3 carbon atoms is carboxylate to form oxaloacetate. Oxaloacetate contain 4 carbon atoms and this reaction mediated by PEP carboxylase enzyme. Number 2, PEP carboxylase enzyme has much high affinity for carbon dioxide than Rubisco and no affinity for oxygen. Number 3, oxaloacetate is reduced to malate. It actually uses NADPH but not stated here, and malate is transported to bundle sheath cell through plasmodesmata. Number 4, inside bundle sheath cell, malate is oxidized and decarboxylated, remove carbon dioxide and then produce pyruvate which contain 3 carbon atoms and release carbon dioxide and also producing NADPH. Number 5, pyruvate enter back to mesophyll cell and phosphorylated to regenerate the PEP by using ATP. Number 6, bundle sheath cell is impermeable to carbon dioxide, so carbon dioxide is retained within the cell in high concentration. Number 7, carbon dioxide enter Calvin cycle in bundle sheath cell because carbon dioxide is higher in bundle sheath cell, make Rubisco to accept carbon dioxide and produce sugar. The sugar is then transported to vascular tissue, which is the vein. Now, can you answer these questions? Number 1, what is the meaning of C3 and C4? 2 points. Number 2, identify the example of C3 and C4 plants. 2 points. Number 3, can you draw an anatomy of C4 plant leaf? 5 points. Number 4, how can a C4 plant minimize the cost of photorespiration? 5 points. The second group of plant that evolved to minimize photorespiration by maintaining high carbon dioxide level using stored carbon dioxide in the form of organic acids is CAM plant. So CAM referred to crassulacean acid metabolism. This plant include the succulent water storing plant type, for example cactus, pineapple and aloe vera. These plants usually live in the desert, and the ideal solution to prevent water loss by closing their stomata during daytime. However, the problem is that photosynthesis can only occur during daytime, and if stomata is closed during daytime and plant need carbon dioxide during the day, how can this plant get carbon dioxide, and how plant can solve this problem? Okay, CM plant also use the exact C4 pathway like C4 plants to maintain the level of carbon dioxide. It freezes carbon dioxide the same way the C4 plants do, except CM plants fix everything in mesophyll cells. CM plants open stomata at night and fix carbon dioxide throughout the night and store the source of carbon dioxide in the form of organic compounds in the vacuole at night. Thus, we call it as CAM pathway. So when the stomata is closed during daytime, carbon dioxide is released from the organic compounds, and the carbon dioxide is then used in the Calvin cycle to produce sugar. And remember, this is really important. The CAM pathway is similar to the C4 pathway, except the site for Calvin cycle. Calvin cycle of C4 plant occurs in bundle sheath cell, while Calvin cycle of CAM plant occur in mesophyll cell. How the CAM plant leaf can reduce the photorespiration using stored carbon dioxide in the form of organic acids? Notice that CAM plant has no structural rearrangement in their leaf, and CAM plant has similar structure as C3 plant leaf. CM plant close the stomata in the presence of light and open the stomata in absence of light. So when stomata is open at night, carbon dioxide diffuse in and oxygen diffuse out. This results in higher concentration of carbon dioxide within the mesophyll cell of plant leaf at night. How CM plant can reduce photorespiration? Use the zoom in picture in the mesophyll cell to explain the process. Okay, during the night, when stomata is open, carbon dioxide is picked up by the enzyme PEP carboxylase, this kind of Pepsi, and attached to the PEP molecule, okay, PEP phosphoenol pyruvate, and this results formation of molecule oxaloacetate. Oxaloacetate is then converted to malate. Alright, malate. This malate is stored in vacuole here until light is become available. Malate aka malic acid is the same thing. And then during the day, stomata is closed. Malic acid or malate will exit the vacuole and releases carbon dioxide. Okay, releases carbon dioxide and then become pyruvate. Okay, pyruvate reconverted back to PEP with energy input of ATP during night. Okay, so pyruvate, it is the same name as triose phosphate. Triose phosphate refer to the pyruvate. Alright, so what happened to carbon dioxide here? Carbon dioxide can incorporated into RUBP by Rubisco enzyme in the Calvin cycle and produce sugar, which is the G3P. And then the sugar produced from Calvin cycle is transported into the vascular tissue. Let's recall back all the discussion for CM plant and how they can reduce photorespiration. CM plants only use mesophyll cell, remember that, to fix all the carbon dioxide, and during the night stomata is open. PEP fixes with the carbon dioxide producing oxaloacetate, and then oxaloacetate reduced to malate. Malate stored in the vacuole throughout the night to prevent pH changes in cytoplasm, while during the day stomata is closed, malate oxidized and decarboxylated producing pyruvate and carbon dioxide. Carbon dioxide is released and used in Calvin cycle, and then pyruvate is phosphorylated by ATP and regenerate the PEP. This results increased carbon dioxide concentration in mesophyll cells and photorespiration is prevented. Can you answer all these questions? Number 1, what is the meaning of CAM? 1 point. Number 2, identify example of CM plants. 2 points. Number 3, how CM plant minimize the cost of photorespiration. 5 points. Notice in this diagram, the CAM pathway is similar to the C4 pathway. With the first step is carbon dioxide is incorporated with the PEP by PEP carboxylase enzyme producing organic acids named oxaloacetate. Oxaloacetate then release carbon dioxide and enter Calvin cycle. Please look at the number. Number 1 is initial carbon fixation. Number 2 is carbon dioxide released to the Calvin cycle. Keep in mind that C4 CM plants are eventually use Calvin cycle to make sugar from carbon dioxide. They have additional pathway in order to increase concentration of carbon dioxide in the leaf. The differences is that in C4 plant, the C4 pathway is separated structurally from the Calvin cycle, whereas in CM plant, the CM pathway and Calvin cycle occur at separate times but within the same cell, which is the mesophyll cell. So C4 pathway minimize photorespiration by separating initial carbon dioxide fixation, which is the C4 pathway, and the Calvin cycle in space, okay, using two different cell. Thus, it is termed as spatial separation. Spatial means space. While crassulacean acid metabolism plant minimize photorespiration and save water by separating initial carbon dioxide fixation and Calvin cycle in time between night and day, but within the same cell. Thus, it is called as temporal separation. Temporal means time. Now, look at the differences between C3, C4, and CM plants. Can you identify 5 differences between them? For example, what are examples of plant? It easy. And then, how many carbon fixation occurs in each plant? For example, C3 only once, while C4 and CM twice. Because C4 and CM, they have additional pathway in order to increase the concentration of carbon dioxide. And then, aspect number 3 is site for carbon fixation. C3 and CM only involve one type of cell, which is mesophyll cell, while C4 it uses 2 different types of cell, which is initial carbon fixation in mesophyll cell and Calvin cycle in bundle sheath cell. And aspect number 4, carbon dioxide acceptors. C3 REBP only, C4 and CM REBP and PEP. What are the enzymes that involved during fixation of the carbon dioxide? C3 rubisco, while C4 and CM rubisco in PEP carboxylase. What about rate of photorespiration? What about rate of photosynthesis? And what about the yields of sugar? You answer these questions. Number 1, what is the type of separation for CM plant and C4 plant? 2 points. Number 2, explain each type of separation. 4 points. Number 3, list the differences between C3, C4, and CM plant. At least 5 differences. 5 points. These are some of the references that I used, and that's all from me. Thank you.